Thank you, thank you. Yeah, we have just finished the exciting presentation sections this morning. So the first speaker, Professor Washer, talked about the biocatalysis, and rest of three will talk about the chemical catalysis. So I'm going to post a few questions for all of you so that we can share their wisdom with all of us. So the first question will be, let me see, I have prepared a few of them. What are the advantages and limitations of using enzyme, that means biocatalysts, compared to the traditional chemical catalysis in industrial processes? Yeah, Xinyuan, you might be starting.
Okay, so maybe, so let's start, okay. So I think the enzyme catalysis and chemical catalysis, the both is very important. So let me think back to maybe 2012, one paper in Nature, so organized by the very famous synthetic biology Professor Qilin and organic catalysis Professor Baron, about the constructive debate for the synthetic biology and organic chemistry. So they show the key the difference. So anyway, so if you are interested, I recommend to read that paper. So for me, so in my point, like the enzyme catalysis, maybe have some many advantage and maybe have some limitation. For example, like enzyme catalysis, so I just show the chemistry, so learn from the enzyme, because enzyme catalysis, sometimes the optimization reaction is very fast. So because more recently, like develop very powerful the directed resolution technique, so developed by the Nobel Prize laureate, the Professor Arnold. So because this strategy, so is very quick to give the enzyme the library. So that means the optimization reaction is very easy because have many catalysts. So on the other hand, maybe enzyme catalysis provide the excellent inactive selectivity control, because enzyme can provide the very well confined carrier pocket. So also enzyme catalysis maybe is very quickly to give them the very complex organic compounds with multiple cancers, multiple carrier centers. So however, the enzyme maybe have some limitations. So I just mentioned maybe the subscope and the reaction scope is very limited. So because I just mentioned the enzyme catalyst with well-confined pocket, so maybe the subscope as compared to homogeneous catalysis, so maybe it's narrow. So maybe some reaction also some subscope. Subscope is very narrow. So I want to say maybe like enzyme catalysis, most carry out in the water. So maybe some reagent, so very sensitive to the water, maybe is very difficult tolerate in this reaction system. So anyway, so I want to say the enzyme catalysis and the homogeneous catalysis both are important. So that means we should have enzyme catalysis have the limitation and advantage, and homogeneous catalysis have the limitation and advantage. So that means collaborate with each other, so to become the powerful tool for the synthesis for the very important products. So this is my point.
Okay, thank you, Xinyuan. Professor Zhou, please.
Okay, 这个刘Xinyuan老师 oh sorry I speak Chinese. Professor Liu already mentioned the both side advantage and limitation of this too. But in my opinion, the enzyme, the biocatalysis has more environment friendship. And another advantage is more specific, it's more selective. Compare chemical catalysis, but the chemical catalysis is much more powerful and can do much more reactions than the enzyme, because the enzyme only can catalyze at the moment natural reactions. But I think both sides need to learn from each other and to address the limitations. And this is the situation already we're facing now. So this is my opinion. We should learn each other.
Okay, thank you. Zhiwei, please.
I think both professors have already expressed very good points on the comparison between like enzyme catalysis and chemical traditional catalysis. I'm not an expert on enzyme catalysis, but I know a lot of the good friends in that area. It seems to be a very hot topic right now, but actually it's not a novel idea. It can go back down like decades. There are even like a lot of good processes currently going on in the industry that rely on enzyme catalysis. But if there is a competition between those two fields, I'm certainly on the team of chemical catalysis. Of course, I'm going to lose. I mean, enzymes are so good at control stereochemistry and radial selectivity and such. But for the long run, I'm thinking, why not limit the space for innovation? There's so much things you can try, like so many metals and so many elements that synthetic chemistry can put together, right? Why not just focus on a very complicated system? And it takes a lot of effort to do that, and some of the time are not really robust. Because we recently, we played around a little bit with like photocatalysis and enzyme catalysis. One of the major challenges is that those things are not as robust as our organic molecules, and sometimes compared to like transition metal catalysts. So I think, I especially like the quote of the founding mother of biomimicry. Learning from nature is not just to copy it. It's to understand the principle and leverage the very core of that, then come up with like entirely new solutions. I'm absolutely for that. Thank you.
Okay, thanks. Thank all of them. Things we have a few minutes left, so I will turn to very simple questions, but might be interested by all the audience. So what is your dream catalytic reactions and why? Xinyuan, start.
Okay, so actually, so I just show my chemistry, so use all developed catalytic system to realize looked very beautiful, the carbon-carbon or the carbon-hydrogen bond formation. So actually, so you can see so our ligand structure very complex, and some reaction use very high catalytic loading, so reaction efficiency is not good. so why we continue to think about so for me it was a dream reaction. So we want to continue focused on the asymmetric radical reaction. So we hope to develop the reaction directly from the easily available the starting materials, like the alkene or the alkanes or very simple alcohol or the amine or even more the biomass, the source, directly give the value added the chiral compounds. So in one step. So also we hope use our develop the ligand really cheap and very easily available and with high reaction efficiency and very low the catalytic loading. So if the OK, maybe provide a very important tool for the synthesis, very important chiral drug and advanced material. So anyway, so have very long time, very long way to go. So this is my dream reactions. Okay.
Okay, good. Qilin, please.
My dream, of course, is we want to develop a universal catalyst. It can solve all the problems. But we know this is just a dream. We cannot realize that. But what we can do is develop a privileged catalyst. Why we say that? Because now we have many reactions, especially in the organic chemistry. Almost an unlimited number of reactions, unlimited number of the substrate. If we don't have a privileged catalyst or ligand, that means we need an unlimited number of the catalysts. That's the task we cannot approach. So this is why I say at least we need to develop the privileged catalyst, which can solve many, many reactions and subjects.
Okay, thanks. Zhiwei, please.
I have so many. Maybe just name two, like methane and water to make methanol, and methane and carbon dioxide to make acetic acid.
Good. Thank you very much. It's time for us to have lunch. So I think I have to stop here. Otherwise, you might not have enough time for your lunch. With that, I'd like to thank all the participants this afternoon and thank all our speakers. And thank you very much for joining this chemistry symposium, and I'd like to close this morning's chemistry symposium.